In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, how are you? I hope you all are fine and enjoying your health and studies. Dear students, I welcome you in Pakistan International School Ties virtual learning system. And you know, this is your class of biology for grade 8th, which is now 9th junior. Dear students, we have started chapter number 4, that is about cells and tissues, and you know that is very long chapter. That's why it will take some time. So you don't worry, inshallah, we are in half of the chapter now. So chapter number 4 about cells and tissues, we have seen lots of things about cells and tissues because these are the basics of the life. So first we have studied the cell theory, then we have seen cell structures. Now we are looking at cell organelles, in which we have studied a lot of cell organelles. Now we are gonna see some more cell organelles, which are lysosome, centriole, vacuoles, and then we will see difference between eukaryote and prokaryote. So let's get started. Before we have seen lots of, we can say, cell organelles, in which very important cell organelle we have studied endoplasmic reticulum, which it is having two types of endoplasmic reticulum, smooth and rough endoplasmic reticulum. So if we see rough endoplasmic reticulum, so that is having connection with Golgi complex or Golgi bodies. Now there is another cell organelle which is having the connection with Golgi bodies, because the vesicles which arise from the endoplasmic reticulum or smooth endoplasmic reticulum, they form Golgi complex or Golgi bodies, and from these Golgi bodies some vesicles modified and then come out of the Golgi bodies. Those are called lysosomes. So you can see here the lysosomal structure. These are very simple because they are single membrane bound organelles. So lysosomes were discovered in the mid 20th century by Belgian scientist Christian René de Dove, and he discovered some single membrane bound organelles present in the cell. So in the cell, these single membrane bound organelles, you can see here this blue one, they are having very important role in our cell's life because they contain strong digestive enzymes. You know, digestive enzyme for like the chemicals, the proteins, the enzymes which are used to break down some substances or break down the food as well. So you can see here in this is lysosomes. When the food move inside it, so with the help of enzymes, it's broken down into very, very smaller pieces. So let's talk about the important function of cell organelle, the very important cell organelle, that is lysosomes. So lysosomes are having digestive enzymes, and these digestive enzymes work for the breakdown or digestion of food which is entered into the cell, and waste materials within the cell, or the waste material which is formed in the cell, that is digested or broken down by the lysosomes. So you can see here the vesicles arise from the Golgi apparatus, and that it is linked, or it is fused with this, we can say any type of vacuole or fragment, any type of phagosome. Phagosome means this is um, um, like a body, a vacuole-like body in which there is food present, and this food particles comes from out of the cell, and then with phagocytosis. Phagocytosis means by eating of the cell, or we can say when it comes into the cell, so it just come inside a membrane, single membrane, that is called phagocytosis, or this is called phagosome. So we can see here, this is lysosome, it's coming here there is in the vacuole, and phagosome there is food, and this just fuse with this one and just enter or leave its digestive enzymes in the food, and the food is digested. Any type of bacteria or um, like any type of foreign particle just comes in the cell, or it is present from the cell, because some waste material is also um, just present in the cell. So it breaks down all type of materials which are unimportant or not necessary for the cell. So during its function, a lysosome fuses with the vacuole that contains the targeted material, and its enzymes break down the material, same as this process which is running here in this animation. Just tell me one thing. So okay, what can happen when a lysosome bursts inside the cell? Because there are not only one lysosome, there are lots of lysosomes in our cell. So in one cell, lots of lysosomes are there that do perform their function of digestion, because they are having a certain digestive enzyme. So tell me, what will happen when a lysosome bursts inside the cell and all its enzymes are released in the cytoplasm? Can you tell me? Just think about it. Yes, and think about it, then I'll ask the answer in the Zoom meeting soon after your lecture, inshallah. Okay, so here you can see from the Golgi apparatus, primary lysosome, the lysosome which is newly formed from here, it is having certain digestive enzyme because it is modified in the Golgi apparatus, and then it is um, moving and just fusing with the vacuole which is having the targeted material. It can have any type of bacteria, any type of waste material, any type of food which is present in the vacuole, and then after fusing it releases its digestive enzyme in the vacuole's material, and then this material just breaks down uh, and digested, and then this lysosomes and release this material out of the cell. Okay, now we will see the next one, the next cell organelle, that is centrioles. Centriole comes from the word center, like it is present in the center of the cell beside the nucleus. Centriole, remember one thing, that it is only present in the animal cell. It is not present in the plant cell. There are such structures which are present in animal cell, not in plant cell, and some which are present in plant cell, not in animal cell. Like what? Chloroplast, like cell wall, they are present in plant cell but not in the animal cell. So centriole is a structure which is present in the animal cell, not in plant cell. So let's talk about this one. Centrioles. Animals and many unicellular organisms have hollow and cylindrical organelles. These are called centrioles. You can see these are hollow from inside. Hollow from inside. There is nothing inside it. If we talk about its structure, so it is made up of 9 triplets of microtubules. Triplet means 1, 2, and 3. microtubules. You remember microtubules we have seen in where? Cytoskeleton, yes. So these are microtubules, you can say three straws or three fluty chocolates. So these are just stick together with some proteins. These three, 1, 2, and 3, they form triplets. So how many triplets are there? 9 triplets are there. 1, 2, 3, 4, 5, 6, 7, 8, and 9. So they arranged in the form like they form what? They form steric-like structure, like star-like structure. Remember one thing, microtubule is made up of tubulin protein. So tubulin protein is present in microtubules. Animal cells have two centrioles located near the exterior surface of the nucleus, and if you see the animal cell, this is animal cell. In the center there is nucleus. On one side there is centriole. One centriole is present. Animal cell, two centrioles are present, and they are formed like this placid structure, like perpendicular, one on x-axis and other is on y-axis. So at the exterior surface of the nucleus, they collectively, like this collective portion of centriole, that is called centrosome. This is called centrosome. So what is its function? We will talk about in the next slide. But first of all, you have to see its structure. What type of structure it is having? It is having steric-like structure in which there are triplets or 9 triplets of microtubules which are connected with some proteins, some more proteins. Okay, so these are called collectively, both are called centrosome. Function of centriole is very important in cell division. What is cell division? We will talk about in the next chapter, inshallah. Its function is to help in the formation of spindle fibers during cell division. Spindle fibers, like spindle-like structure, like thread-like structure, they form. This is centriole, if you see, or this is called centrosome. Look, and then it is duplicated. It becomes 2, and 1 move to other side of the nucleus, and they form spindle fibers. These thread-like structure, which you can see here, these are called spindle fibers. These are fibers-like structures. Actually, they attract these chromosomes which are present in the nucleus. This is a little complicated process, but when you go to chapter number 5, in which you will see mitosis and meiosis, two types of cell division, then you can understand it well. This is eukaryotic cell, but animal cell it is. In the center, there is nucleus. There is one centrosome. But when the cell is about to divide, it just also duplicates and becomes 2 because one has to move on the other side. And then it forms spindle fibers. The function of spindle fibers is that they is prolonged from one side to another side, and these chromosomes attached here on this one, and the other spindle fiber attached to the same chromosome from the other side, and they pull the chromosome to both of them towards themselves. Then chromosome break apart from inside. When chromosomes becomes half, nucleus becomes half, and the cell divides from 1 into 2. This is very important part of the cell division. We will see in chapter number 5. Don't worry about this. Inshallah, you will have the best concept of this, how the cell divides. Okay. Centrioles not only helps in the cell division, but it also helps in and involved in the formation of cilia and flagella. What are cilia and flagella? I have mentioned before also when we have seen cytoskeleton. Cilia are very small hair-like projections, hair-like structures which are present around any body, any living creatures. This is paramecium, which is unicellular, and it is having, you can see at the corner here, these are thread-like structures which are moving, and these cilia helps in the movement of the cell. This is paramecium unicellular eukaryote, which is moving with the help of these cilia, and they are all around this. Cilia are so many and very small in length. Other structure is flagella. So if you see, flagella is longer thread. This is euglena, and the real euglena, which just you can see from the pond water or from the river water, we can collect them. So this is a euglena, and it is having these thread-like structures. So you can see that flagella is longer rather than a cilia, and this is just less in number but longer in length. So they both are used to help in the movement of the cells. This is one cell, that's why for the movement something is needed. So here in animal cell, that is used for the formation of spindle fibers. In the cell division unicellular, they also help for the movement in the form of cilia and flagella. Okay, the next one, very important structure as well, this is vacuole. Vacuole, you can see here, the animal cell, eukaryotic animal cell, this is lysosome, the blue one, and these are smaller, which are coming from the Golgi apparatus, these vesicles, these are said to be vacuoles. And vacuoles, if we talk about a structure, vacuoles are single membrane bound organelles, and that's why they are also present in unicellular organisms. They are fluid filled organelles. They don't have digestive enzymes, but they are full fluid filled membrane, single membrane bound organelles. So if we talk about in animal cell vacuoles are very smaller in size, but they are so many in numbers. So that was actually, this is lysosomes, but here these vacuoles are also present. There are so many in numbers and smaller in size. They are having some food material, some fluid, which are stored in the cell, which are required by the cells after some time or for their energy production. In plant cell, there is somehow a difference that in plant cell, there is one large vacuole. You can see nucleus is pushed aside. Nucleus is pushed aside, and this pushing is due to this large vacuole. When the plant is very small, so many small vacuoles are present in the cytoplasm, but when it becomes larger, the small molecules fuse together and they form big, large molecules which come in the center and push the nucleus at a side. And this vacuole is having lots of fluid in it, water in it, salts in it, some food materials inside it. And the cell in this state becomes turgid when it becomes very large, and it absorbs lots of water and fluid and salts due to what it becomes turgid. Turgid means hard, like when we blow in balloon too much air, then up to after that it will burst if we blow it more. Like that, it is having the fluid inside it or liquid and water, so that it must be turgid. This turgidity or this stiffness of this vacuole is very important for maintaining the shape of the plant cell, because it has to maintain the environmental conditions also outside outside environment. That's why. So in higher animals, in higher plants and larger plants, vacuole is one, but in smaller plantlets, vacuoles are too many and smaller in size. They become fused, and then they just transform into the large vacuole. What is the function of that? That it takes in and stores the material from the outside in the form of food vacuole and digests the material within with the help of lysosomes. We have talked about a lot about the lysosomes before. If we talk about vacuoles, so some unicellular organisms use not only food vacuole, but there are some other kinds of vacuoles. Those are called contractile vacuoles. If you see this eukaryotic animal cell, it is having, like, when the cell is eating, this process is called phagocytosis, and this is phagocyte, which like it is formed now, and a phagosome actually. So it because this cell membrane just twisted around this cell, this food, and then it becomes a smaller organelle. This is called food vacuole, and this membrane joins from here. This is called phagocytosis process. So here it becomes the food vacuole. Now it just fuses with the lysosome, and lysosome just releases its digestive enzyme inside it, and then it breaks down and digested, and then exocytosis is done. Exo means out, cytosis means cells from outside of the cell. This all the undigested material just removes out of the cell. This is food vacuole, but in some unicellular organisms, there is contractile vacuole is also there. Contractile comes from the word contraction. So when this vacuole contract, you can see in Paramecium, this one and this one, two contractile vacuoles are there. So when they contract, look, this one and this one is contracting, so it eliminates the waste and extra water from its body to maintain the water content in its body, because it also always lives in the water. So food vacuole also present, so many food vacuoles, but contractile vacuoles also present for contracting out the water and waste from the body. Let's talk about very important topic here, difference between prokaryotic and eukaryotic cells. We have seen it before in the chapter number 1 also. Very important, both are having prokaryotic, which are much similar than the eukaryotic cell. So somehow it is similar, but somehow it is different as well. First difference which we'll talk about, that is about the nucleus, by which they are said to be prokaryotic and eukaryotic cells. So this is prokaryote and eukaryote organism. Pro means first, karyote means nucleus, eu means true, and karyote means nucleus. Like in prokaryotes, they arise from the origination of the nucleus before that. And eukaryote, these organisms are having true nucleus. In that, true nucleus means it is having membrane bounded nucleus, in which they are it is having what, chromosomes. But here in prokaryote, a prokaryotic cell, the nucleus is not present properly, but the DNA material or genetic material that is floating inside the cell, in the center of the cell. So the place where the chromosomes and DNA just floating in the center of the cell, that place is called nucleoid. Now, this is the region where the nucleus is present. Actually, proper nucleus is not present. So some other organelles which are having membrane bounded membranes, double membranes or single membranes, they are present in eukaryotes. They are not present in prokaryotes, because prokaryote means they don't have membrane bound nucleus, so it does not have any membrane bound organelle also, like mitochondria, Golgi apparatus, endoplasmic reticulum, etc. And membrane bounded organelles are not present in prokaryotes, but they are present in eukaryotes. Let's talk about ribosomes. So ribosomes, if we see, ribo comes from the ribonucleic acids, like from RNA. Ribonucleic acids, because it is a combination of ribonucleic acid and protein, so means body. So it is the body which is having ribonucleic acids. So ribose of eukaryotic cells are larger. This is eukaryotic cells. Its ribosome is larger. You can see the difference between the size also. It is having larger ribosome. It is having smaller ribosome. Prokaryote is having smaller ribosome because it is more smaller than the eukaryotic cell. Size, if we talk about, so eukaryotic cells are on average 10 times larger than prokaryotic cell. You can see this is eukaryotic cell, this is this is prokaryotic cell, and this is eukaryotic cell. So you can see the difference between the sizes of these two cells. So the eukaryotic cell is 10 times larger than prokaryotic cell. Let's talk about cell wall. So cell wall of eukaryotic cell is made up of cellulose, like in plants, or cell wall in fungi, that is made up of chitin. Chitin is all, fungi is also eukaryotic, but in prokaryotic, the cell wall is made up of peptidoglycan, like in bacteria. You can see lots of bacteria here, which are prokaryotic in nature. And peptidoglycan is the polymer. Mer means molecule, poly means many. So that is the larger molecule in which there are very small molecules are there. So small molecules get together and they form larger molecules. It means lots of amino acids are there and sugars are there. If sugar is present, that's why they are called glycan. And if amino acids are present, that is called peptide. So that is peptidoglycan. So here we will finish today's lecture. We have finished and we have completed all the set organelles, dear students. So you must learn all of them. You have to write the question answers and the topics with the diagrams. All the topics you have to write on your notebook and try to make the diagrams. Inshallah, we will see you in the next lecture. So up to right now, that is difference between prokaryote and eukaryote. You have to complete your notebooks as well, and then we will see some exercise and question answers, and then we'll see the next topic, inshallah. Till then, wish you all the best. Allah Hafiz, best of luck.